So welcome everybody, and thank you very much for joining us today. I can see we've got a lot of people online already, and there's a few more just logging in now, but we'll get started and catch up. So the title of today's webinar is Automated Organoid Counting, and we'll be using the DeNovix CellDrop Automated Cell Counter for this. Little look at the agenda for today. So for those of you who aren't familiar with CellDrop, we'll give a brief introduction to that technology, just so you know a little bit about the instrument that the counting will be done on. We'll take some time to talk about the challenges of counting organoids, tumor spheres, and spheroids, and how the complexity of these samples affects the ability to use traditional automated systems for this application. And obviously talk a little bit about how the DeNovix solution has solved these problems, discuss the machine learning technology that we've used and how we've implemented that on CellDrop. And the majority of today's session really would be taken up by a live demo. So we'll switch to the lab in Wilmington in Delaware for a live demo of some organoids that we have prepared in our lab. And we'll also then put some time aside for a Q&A at the end of the presentation with our scientist who'll be giving the presentation today, Grace Emin. So in terms of questions, at any point during the proceedings today, if you have any questions, please put them into the Q&A panel that you'll find on the Zoom interface. We have some of the application support team on the line. They'll answer some in real time, and then we'll take some to the live discussion as well at the end. So a little bit about DeNovix for those of you who aren't familiar with us. We were founded in 2012 by Fred Keelhorn, who was one of the original founders of nanodrop technologies. And our ethos and our mission really was to make innovative and unique laboratory equipment for the life sciences, and really to look at genuine pain points for scientists and try and solve them in creative ways. So since 2012, we have launched a range of microvolume spectrophotometers and fluorometers, as well as the CellDrop that we'll talk about today. And we have thousands of these instruments placed globally. And if you look at if you look at Google Scholar, we have thousands of citations using our instruments. And our instruments are also some of the most reviewed and respected instruments on the market, reviewed by scientists all around the world. Today's speaker is Grace Emin, and Grace is a member of the DeNovix application support team. Grace has a master's degree in biological sciences, which had a focus on biomaterial engineering and disease modeling. And she's been a key contributor to the development of the Organoids app on the CellDrop. And she's going to be the one to take you through the application today. So I'm going to hand over to Grace, and good luck.
Hello, everyone, and thank you, Andy, for that introduction. Before jumping into things, we're going to start off with a poll to hear about your experiences. Specifically, we want to know about your primary challenges when quantifying organoids. So is it accuracy, clumping and overlapping, variability, time and labor, or is it something else? And you can drop it in the Q&A panel. So we'll give a couple of seconds. All right, so I'm seeing clumping and overlapping and accuracy issues. Well, the good thing is I will be addressing all of these, and you can see how we are here to help with that. All right, so the device that I will be demoing and discussing with you today is our CellDrop FLI. So some key features to get us started. We are able to count cells without slides or no consumables thanks to our direct pipette technology, allowing you to save time and money there. So of course, in addition to bright field, we have dual fluorescence. So this allows for a wide range of assays, applications, and sample types that we are compatible with. Most recently, we have been diving into machine learning apps, such as the Organoid app. And all of the data is reported with an easy-to-use software, and you'll see the speed and ease of use when we go through our demo together. Not only that, but the device comes with preloaded apps, free software updates with new added features and applications, and we have multiple exporting options. So what are organoids? So 3D cell culture models mimic in vivo conditions far better than traditional 2D monolayer cell culture. So for examples, just to start off, they are normally classified on size, density, and cell type, but a bit of a disclaimer is that the requirements for the classification of these groups may differ from lab to lab, you and I, but the take-home message is that we can accommodate these and related structures as long as they're within that 4 to 400 micron dynamic range. So for some examples, there are spheroids, which is a term for any simple 3D structures multicellular. One layer up in complexity are tumor spheres, so they are tumor cell based, normally a bit more dense and still sphere-like. And then being the most representative of in vivo conditions are organoids. And so they are dense and, as the name suggests, are organ-like structures. So the cells can have polarity, the cells can interact with one another, and they can even interact with their environment. Why are organoids important to the scientific field? Some of many applications would be drug testing for rare diseases, studying regenerative medicine and new tissue engineering approaches, can help you study disease mechanisms and disease modeling, cancer progression research, and what is very cool is that this is becoming an alternative to animal models, which ends up being a less expensive alternative. So there are challenges with counting these complex structures. So you can see to the right is an image from some in-lab grown tumor spheres. And so you can see very clearly the irregular shapes and sizes. They are not uniform. There can be the clustering and overlapping of these structures. There might be variability in density, which as a result of bright field microscopy will result in variability in brightness. Depending on the integrity and health of your sample, there might be debris, dissociated objects, and the automatic cell counters in the field are optimized for single cells, not these structures. There can also be subjectivity of manual counts, and there's this lack of standardization. So there's no universal criteria for what defines, for example, a spheroid. There are different tools or methods used. There might be variability in protocols. And so this also makes it difficult to compare data across studies or labs. Some of many impacts of inaccurately counting these structures is that it can compromise the reproducibility, reliability, and the accuracy of the data collected. There might be the constant need to repeat experiments, which will result in delayed research project progress. This will waste reagents and time, might result in unnecessary troubleshooting. And so, for example, if we're looking at the negative impacts for drug delivery, you might get inaccurate dose response, toxicity studies, false positive and negatives. And so these are just some of the many negative impacts that we are here trying to help remedy. How are we tackling this? We are using machine learning to count these structures. And so we are using adaptive algorithms trained by DeNovix scientists. So we are scientists working with you scientists. And so we've done all the hard work behind the scenes so that all you have to do is load your sample and measure. So it's very important to note that there is no training at all on your end. So because we have scientists that have worked on this model, there is this robust object recognition. So we're able to discern between organoids, dissociated objects, and debris. The nature of our software is that there's the ability to have user-defined parameters and protocols. So you have the ability to customize and tailor the device to work best for you and your samples. Bit background of machine learning in case it's new for you. So we took image sets and we told the software what class each object is to be in. So in the case for us, whether or not it's an organoid, dissociated object, or debris. We'll get an initial algorithm that is not perfect. So then it is then corrected by a scientist using different sample types, different conditions. So we're retraining it over and over and over until the algorithm results are accurate for the application. So in this slide example, machine learning was used to identify different features to allow for the classification of whether or not it is a cat, dog, or bird. For us, our algorithm model is looking for specific features for organoid, dissociated objects, or debris. And so this is a respected method as machine learning is used for a variety of image-based applications out there. How the CellDrop works without using any slides is that there are two pieces of optical grade sapphire. They are parallel and permanent. And when they come together, they create a chamber that functions similarly to a hemocytometer or disposable slide, which you might be more familiar with. Capillary action will pull the sample into the field of view. Surface tension will keep it in there. And when you're done, you'll wipe the sample surfaces with a Kim wipe and there won't be any carryover. The nature of the device is to be self-sufficient, so there's no calibration required. And since we have such precise control of the chamber height, we can achieve different chamber heights to accommodate different sample densities. So for example, for organoids, we have the 200 and 400 chamber height, which is different from that traditional 100 micron. And so counting cells without slides is what we are first known for and quite proud of. And so we're incorporating environmentally friendly innovations in workflows. To walk through the organoid workflow, you'll load the sample like usual. You will check the focus for the first time loading this sample type. The device will remember your previously used settings, so you will not have to do this every time. When you tap count, you'll get a machine learning powered analysis in under 20 seconds. You'll have the results, so you'll have the result image to the right, data portrayed on the left. You have the option to now or later, because data is automatically saved on the device, to export. You'll wipe the sample surfaces clean with the Kim wipe, and then again, you're ready to load your next sample. So of course, we can do the easy applications such as basic cell culture, routine counting, and bright field-based counts, but we're also able to do the more challenging and cool applications out there. So another machine learning app we have is hepatocyte counting. With dual fluorescence, we can measure the nuclei extraction efficiency. A more recent assay we've released is apoptosis assay. So we can look at the early and late apoptotic cells and then live and dead. We have the dual fluorescence viability assay. So this is great for primary cells because any debris, non-nucleated cells, platelets will be completely disregarded from the total cell count. We have a higher magnification device as well, the FLXI, so we can accommodate smaller cells in that 2 to 20 micron range, such as stem cells, so we can measure their viability. And then also we can work with yeast cells, so we can measure yeast vitality, which is not the same as yeast viability. So a summary of the key benefits before we hop on over to our demo. You have the ability to save cost and time since there are no consumables, and you will get machine learning counts in seconds. We have sample versatility, so we have the organoids, spheroids, tumor spheres, and structures alike. But we also have a wide range of other apps available now, and we're in constant development, so we always want to know what you would like to see next. The software is easy to use and allows for you to have high reproducibility between users. And so this will also help remove subjectivity from analysis. So this app can serve as a method to help standardize your data collection and analysis for this difficult sample type. So add some consistency and reliability to the workflow. So before jumping into the demo, I'll make time for any questions that can be dropped in the Q&A panel.
Thanks, Grace. We've had some questions come in already, so let me give you those ones. So first question was regarding the CellDrop. How can you tell if the sample chamber is clean when you've removed a sample? And can you use a disposable slide if you need to as well?
So the field of view is live. So after you're done cleaning those sample surfaces, you'll be able to see if there's any debris or lint left behind. And yes, we can accommodate slides. We have a slide mode. However, remember, that is with the traditional 100 micron, which may not be able to accommodate all of your organoid sample types.
Great, thanks. Somebody was asking whether you have a minimum and a maximum size of organoid that the machine learning algorithm can effectively recognize.
So our dynamic range of organoid size would be 4 to 400 microns. And you also have the ability to gate out certain diameters with your results.
Okay, great. Another question that's come in is just whether there are any additional reagents that need to be purchased to stain the organoids.
Nope. So this is bright field based and there are no assays required. So you could take a sample from media and then just load it on the sample for counting.
Perfect. And there's a question that maybe I'll take here, Grace, is will the Organoid app be available and usable on previous versions of the DeNovix CellDrop? So the answer to that is the Organoid app is specific to the CellDrop FLI model. So if you have that, then the app can be purchased and used without any modifications. If you have any other model, we do have upgrade paths available, and we can just discuss how best to get you up and running with the Organoids app. And they're all relatively painless. So if you have another model, please do get in touch with us either in the chat or after the webinar, and we'll be happy to discuss how to get you the Organoids app in your lab. Okay, I think that is it for now. If anybody does have further questions during the next part, please add them to the Q&A, and we will put them to Grace at the end of the demo. So, thanks, Grace.
Of course. All right, so hopping over to the demo here. So you're going to see this is our home screen. And you'll see that here we have our Organoid app. These are just the other apps that we have also available at the time. Going to tap open. And so this is a live field of view. So you'll know if there's any debris left behind or you need to do another cleaning. So the sample that I have are Enteroids, and so they are a type of organoid. And so these were maintained in DMIME F12 media, and they were in a non-attachment flask, and I just took these from tissue culture, so nothing fancy there. Just some tips and tricks for counting these structures is you're going to want to make sure that you're pipetting up and down lightly in between your counts of your aliquot to help with reproducibility, making sure that sample surface is clean, and also a good reminder that there are no assays required, so no additional prep beforehand. So the first count that I will be doing is I will go and just show you how quick and easy and fast it is. And then the second time, I will go through more of the fun features and nitty-gritty of the counts. So for those who might be, this is your first time seeing a CellDrop, a bit of an overview. We have the two pieces of optical grade sapphire. So we have the upper and the lower sample surfaces. When they come together, they create that chamber, much like a slide or hemocytometer. And then you'll pipette on either side of the groove. So there's one here, one there. And so I'm a lefty, so I pipette on the left side, but the right side is just as fine. So I'm just going to lightly pipette up and down my sample before pipette it. So just doing that gently. And then I'm going to pipette like usual. Get a live preview. And so you can pinch and zoom. You want to make sure that your sample and all of the cells are settled and they no longer moving. And this is also a great little QC step. See the integrity of your sample and how it's doing. Once it is fully settled, which takes normally a couple of seconds, then you're ready to tap count. You're going to get a machine learning powered count in under 20 seconds. And so this is bright field based. So it's going to take an image in the bright field channel. And then it's going to use that machine learning algorithm that we trained. You'll get the result image on the right, and then you'll have more data on the left, such as the densities. So this is also, again, pinch and zoom. So we can take a look at how well it's doing with actually selecting all of the organoids. So it's quite easy for me to see, but it might not be for you. But the organoids are outlined in blue. Dissociated objects are in yellow, and if you'd like to toggle on debris to see as well. So you see these objects are not being included in the dissociated. So any debris you don't have to worry about influencing your counts as well. So you can see how unusually shaped that is, far from irregular. See some other fun ones we have as well. Again, see how accurate all the different shapes are being very clearly outlined. Okay, so you have the option to name your sample here. You could have also named it on the Count tab. And so there's also the ability to do some fine tuning. So say that you know that you will only count an object as an organoid if it is at least 50 microns in diameter. So I'm going to move there is a live gating here. So I can go to 50. You'll see here you have the ability to apply gating to results. So if I tap that, you'll also have the ability to do save settings to protocol. And now I can save these settings to a protocol, so I don't need to do this gating or any other protocol changes again. I can just recall this. So I'm going to name it the sample type. Save as. And then I'll show you when we do our second count that this is now already being plugged in and it's already been called. So some other fun things we have. So you have the option to export now, or you could export later. But just to kind of show you what that looks like, we have different data we can export. So it can be the raw PNG images, CSV, or PDF. So say I just want to export the result image for now. Then you see some of the many options we have. So USB is what I have right now, but it can also connect to internet for email, network folder, and network printer. You see on the left, we have some data such as the organoids and dissociated objects density. Then you also will get the mean diameter of your organoid and then the % dissociated cell count. Sweet. All right, so I'm going to do the next count here. So all you need to do when you're cleaning in between counts is you're going to wipe the sample surfaces with a Kim wipe. So I'm just folding mine like so. And a firm wipe of the upper and lower sample surface. So this is made with optical grade sapphire, so you don't have to worry about breaking or scratching it. So I'm going to wipe the upper and the lower. Lower it so. Next count. And any little bits of debris that might be left behind, again, it's going to select it as a piece of debris and it's not going to be considered into the next count. So again, just like I did before, lightly pipetting up and down my aliquot. And then now this is a good time to say that we see that our protocol has been recalled right here. And you can make a protocol for every sample type. There is no limit on the number of protocols you can make. So this is one we made together, so that one's being recalled. Another thing is that you'll always know how much of your sample to pipette. So we are currently in the 200 micron chamber, so I have 20 microliters of sample that is to be pipetted. But for example, if we were to use the 400, that number would change accordingly, and then we would have 40 microliters of sample. All right. Just loading my sample again, just like last time. See the structure again, live field of view. Now, say this was your first time loading this sample, and you want to make sure you check the focus just for the first time. I didn't have to before because I already had done this, but just to kind of walk through, you'd see here if you tap focus, you can change accordingly with the coarse and fine adjustment, and a little bit of a indicate, a little info button is that we want to make sure that we're focusing where there's a bright white center and a dark outline of the cells, which you can see. So I'm just going to make sure that they're all settled. Now I will name the sample here, so I'll just name it as well, and you can see it's carried over. Tapping count. So again, same machine learning algorithm is being used in under 20 seconds. You'll get the same data portrayed as last time. This is a great reminder to think about 20 seconds versus the minutes or hours it might take for organoid counting, depending on how many samples you have to count. So we'll look again at what's being selected just to show you the crisp outlines again. Let's see here. You're like, all right, this isn't being selected. That's because it's being selected as debris. Sweet. So some other things we have is that we have different ways of portraying the data. So say I want to go to the graphs. Say that I wanted to look at the organoids by size, and I wanted to look at both my samples. Then you can actually have more than one count on a certain graph. So there's more than one way to show your data, and you don't have to select all of them. You can select a few. These are just some we can do by organoids by size, percent associated objects. So multiple ways of exporting. Again, export like I would here. And you see it recalled that gating that we did together from the last time. Sweet. So I'm going to clean the sample surfaces, and then I'll hop on over to the data app just to show you where you can recall all of this. Clean this with a dry clean again. Data app is right here. And so the minimum amount of filtering that you have to do is just by app. And it's going to show you all of the counts that you've done. So I'm going to scroll down, organoids, search, and you will see here, these are the samples that we did together. So say I want to export both at the same time, going to select both of them. They're going to put it on a single report, pop on over here. And then here is great because then say I want to export both of these. Now I do export, and again, the same setup. So I want the result image, and again, the same thing. So then also another thing is that say you forgot to name your sample, you can do it here as well, and it will carry over when you export your data. Sweet. So I will hand this back on over to Andy to help with any other Q&A.
Thanks, Grace. But quite a few questions have come in, so so brace yourself.
I'm ready.
Okay, the first one was somebody asking about whether the CellDrop can operate in a kind of regulated lab under 21 CFR Part 11 regulation.
Yes, we're 21 CFR compliance and GMP compliance, and so we have IQ/OQ packages as well that can be purchased.
Perfect. There's also a question about whether you can assess the health or viability of the sample using this app.
Yes, so that percent associated objects serves as a little QC step. So you can kind of get an idea of how many of these cells are no longer maintained in those organoid structures. And so it's shown in the field that if they're maintaining those structures, they're happy and healthy.
Okay. Do you have to use 20 microliters of sample, or can you lower the count or lower the amount?
So the minimum amount of volume would be 20 microliters for that 200 micron chamber height. And this is important too, just for the size of the organoids. You want to make sure that they can actually pass through the chamber.
Okay. We also had someone ask whether when you have the app in your lab, do you need to train the algorithm with your own samples? And if so, how many samples will this take?
No, so we did all the training. We've done many different sample types. And so when you have the actual app, there's no training at all. You just load the sample and count just like I did here with you.
And is it necessary to create and save those custom protocols for different organoid types that we use?
Nope. You can just use the default protocol and you can do that each time. Not required.
Okay. And there was a question about export formats. I know you went through exporting at the end there. But someone was asking about integrating into lab management and other analysis software. Are the formats compatible?
Yes, so you can export where LIMS compatible, and then if you want to do maybe image day analysis, you can export the raw PNG images.
Will we be able to provide a sample prep protocol for use with the Organoid sample app on CellDrop?
So the great thing is that, like I mentioned, you can just pull from the media and load the sample. So we don't necessarily need to do any prep beforehand. I pulled my sample directly from a flask.
Excellent. Probably one that's a little bit related to the sample volume question that we had. But there's a question about, can you adjust the chamber height in the Organoids app to count objects that are bigger than 200 microns, for example?
Yes. So you could create another custom protocol using that 400 chamber height, and then you would pipette 40 microliters of sample. And the chamber height will change accordingly. That upper arm will move.
There's also a question that came in about reproducibility.
Mm-hmm.
And then essentially, what is the reproducibility? And if we run the sample multiple times, will we get the same result?
So if I understand your question, if you have the same image and then you keep going back and doing next count without moving, so you're going to get the same results. So you need the same count each time. So if you have an aliquot and you're pipetting up and down lightly, new aliquot each time, there's great reproducibility.
And we will should be having we'll be publishing a technical note on this as well. So there'll be an additional resource for you available. So for pathogen, some organoids, there will be both single cells, cell clumps, and whole organoids. Can we have the software give a total count for all of those?
So the two groups that we have are dissociated objects, which would be anything that would not be considered an organoid. So may that be bits of smaller bits of the organoid or single cells. Anything that's not an established organoid is in that category. And then you have the organoid group.
Okay. And all of that data is exportable, of course. You can view them in Excel.
Yes.
Okay, what if the organoids are very crowded and overlap each other? Can it still count accurately?
Yes, so the different samples that we did the algorithm training for, there were samples that were very dense that had a lot of clumping and the organoids were close to one another, and then we had samples that were very sparse. So we did all the different densities and concentrations. So we can accommodate those clumpy and highly concentrated samples.
So there was a question about, it looked like there were single cells that weren't on the focal plane. Can they or should they have been counted?
So sometimes debris, when they're not in the correct focal plane, can actually look like a cell. And so if it's like a debris that's higher or lower in the focal plane, it might give the illusion it's a cell, but it's actually debris. So, and then if you think about it, if there are, say, 1,000 objects or cells or dissociated objects in the field of view that are being counted, one or two we're not too worried about. It's not statistically significant.
About whether we are able to count the organoids without retrieving them from the matrigel, would they have to be in solution?
So we've done a lot of different trainings of different sample types. So you're more than welcome to try, but what is important is that it can be still pipetted. So depending on how viscous your sample is, that might in fact affect the pipetting. But if it's able to be pipetted, it's definitely worth at least a try.
Okay, so just to clarify, the organoid count is the number of organoids and the dissociated cell count is the number of dead and non-viable cells. Is that correct?
So it's just any object that is not maintained in that structure. And so if you want to be extra strict, you can lower the gating size as well if you really want to make sure those are the only objects. But yes.
Okay. I think that is all the questions, unless I am missing any. So if there are any questions that we've missed, we will go through the list after the webinar. And if you've missed anything or if we promise to follow up, we'll certainly get in touch with you to make sure that you have any answers. If we've missed any coming through in a flurry, that they were at the end. But I think that's it for questions for you in a minute, Grace. So thank you very much.
Of course.
Excellent presentation. We appreciate it. So just to summarize and just to conclude really the webinar for today, the Organoids app is now available for purchase on the CellDrop Fly. So you can contact us at DeNovix, or if you're not in the US, please go to your distributor, your DeNovix distributor. If you don't know who that is, then get in touch with us and we'll put you in contact. We have more information on DeNovix.com. So please take a look there for technical content and for other content regarding CellDrop. We do, in the US, we have a free trial program. So if you're not a CellDrop owner at the moment, please, if you would like to trial one in your lab, we have a program where you can get one there for 5 days. And one of our apps team, like Grace, will guide you through the setup and the training for that. So please do visit DeNovix.com and sign up for a free trial if you'd like one. If you are a DeNovix customer and you have a CellDrop fly at the moment and you'd like to try the Organoids app, please do just get in contact and we can organize that for you too. So finally, thank you very much for attending. We appreciate your time today. You will, after the webinar, get a survey that we'd really appreciate it if you could take a couple of minutes to fill that in and let us know your thoughts. We will also share a recording of this with you. So once we have a chance to process it, we'll send out a recording by email for you. And as I mentioned, DeNovix.com has a ton of resources. Please feel free to go on there. And if you want to talk to a scientist, want to talk to either Grace or one of the team, the applications team, there is an option to speak to a scientist on our website, so we can arrange a call if you have more in-depth questions that you'd like to discuss with one of the team. But I think on that, we can say thank you very much for attending. Thank you very much, Grace, for a really interesting presentation. And have a great rest of your day.